Okay, thank you, Bill. And the next speaker is Sergey Grinshpun. He is my colleague from University of Cincinnati, and he will be specifically talking about how we can control exposure to biological aerosols. So Sergey, please.
Thank you, Tina. First, I would like to thank the organizers for allowing me to present. The talk will be focused, as Tina said, on the mitigation exposure, specifically in relationship to biological particles. The outline, I will introduce a few measurable characteristics of biological particles which have a relationship to exposure mitigation. Then we'll talk about the two major approaches for indoor air purification when it comes to biological particles. One, to just reduce all the concentration. Particles may be biological or non-biological. The other one is targeting their viability. Then we'll be talking about indoor purification methods, some traditional ones like filtration as well as novel methods. And then I will end up with the respiratory protection. In terms of measurable characteristics, regardless whether we're dealing with biological particles or non-biological particles, one important characteristic is concentration if we want to mitigate. Now, we also differentiate concentration particle size selectively. This particle size discrimination allows us to target specific biological particles if it comes to viruses, which are smaller, bacteria, which are larger, fungal spores, which are further larger, and so on and so forth. One thing which is specific to biological particles, what we measure is the viability or percentage of particles which are viable. What we do, we use a culture-based method and essentially measure culturability when incubating microorganisms collected on agar plates and then counting colonies. And then there are other ways of assessing exposure to biological particles, including looking at allergens, antigens, cell wall, membrane materials, and so on and so forth. Now, the size ranges for different groups of biological particles. It's basically, I will start with viruses, which are the smallest creatures, and their size ranges from about 20 nanometers or 0.02 micron in this scale to about 200 nanometers. Bacteria are larger and occupies about an order and a half, starting from about half a micron and almost up to 10 micron. Here is fungal spores in the same scale. Here is pollen, which are much larger particles, and for them, the air purification may not be such an important issue because they simply settle, gravitational settling velocity is pretty high. And then other groups such as aggregates, bacterial and fungal aggregates, microbial fragments that may actually be very health relevant, as well as animal allergens that occupy about 3 orders of magnitude size-wise. Now, in terms of exposure mitigation, first, we're looking at indoor air purification, and we can do it using a freestanding air purifiers, which are more and more popular in rooms, or through a traditional HVAC system, which most of us have in our homes. Now, respiratory protection is actually a legitimate way of mitigating the exposure to biological particles in case we may not be successful in cleaning indoor air. Now, there are two approaches, as I mentioned before, to mitigation of exposure to biological particles. The first one, you take all the particles, you don't care whether they're biological or non-biological, whether they are viable or non-viable, and you try to use a method, let's say filtration, just to reduce the concentration with the hope that this reduction will be across the board. The other one, if you specifically target viable airborne microorganisms and you want to reduce the percentage of particles which are viable for obvious reasons. Now, within or in the frameworks of the first approach, there are many methods which are being used. A traditional one is filtration. You have heard enough about filtration, so I will not go with a lot of details about that. The other one is electrostatic precipitation. What we do in this case, we charge particles before they enter the rather narrow channel, and then you introduce these particles to an electric field, and the particles are collected by this electric field depending on what kind of charge they may acquire, and this in turn depends on their particle size as well as their biological characteristics. There are other methods which are being used, and I would say increasingly used, for indoor air purification or for an illusion of indoor air purification. So among them are ion emission that may or may not be efficient, and ozone generation that I will talk a little bit later, that happen to be not efficient for the particle concentration reduction as well as their viability reduction. Now, just one slide on air filtration. Again, you heard enough about air filtration. What I want to emphasize here, that even if you look at the particle deposition on one single filter, you're not talking about the multi-layer filter, just one single fiber. This is a non-monotonic function of the particle size. And the reason for that is the larger particles are deposited due to inertia primarily, and the smaller particles are deposited due to diffusion, and this mechanism works basically based on a different a function of the particle size. So that means that there is always a most penetrating particle size to any given fiber. The size, particle size, where the efficiency is the lowest. And obviously, you can calculate the efficiency of the entire filter when you have the characteristic of these filters and this EF efficiency of the fiber. Depending on the number of mechanisms involved, such as diffusion and inertia, it's all calculatable. The most important thing here, that this most penetrating particle size, if we come to actually typical filters that we use for indoor air filtration, regardless whether it's biological or non-biological, will fall in the range between 0.1 and 1. And you may remember that this actually corresponds to larger viruses and smaller bacteria, or maybe most of bacteria. So very, very small viruses may be successfully filtered out because of the diffusion. The actual problem with the filtration, it's not a problem, but limitation, comes when we actually intervene in this particle size range. Now, pros and cons of air filtration. High efficiency, that's probably the most highly efficient one, and it's true for viruses and bacteria with the grain of salt that I mentioned before, certainly for fungi and pollen. The most challenging particle size is between 100 nanometers and 1 micron. Now, the clean air delivery rate of a freestanding air purifier, if it's a good one, will be in hundreds. Now, the negatives, higher power consumption, the operation cost issue, because you need to replace filters. If you want a good filter, it's a higher cost of replacement. The noise problem and the limitation associated with applicability in confined spaces. Now, here's electrostatic precipitator. I already mentioned the principle here. So the method was evaluated and happened to be reasonably good, or having a good potential for bacterial vegetative cells. The spores are hardy, both fungal and bacterial spores. So we investigated this in the University of Cincinnati with Gedi Mainelis as a primary investigator. And for spores, it didn't happen to be very efficient. Now, the power requirement and noise problem are not limiting factors for electrostatic precipitators, unlike filters. And obviously, they have a lower operational cost, no filter replacement needed. At the same time, I have to admit that they are not commonly used for removing biological particles at this time. Now, unipolar ion emission. The principle is rather simple. You have an ion emitter, you emit unipolar ion, let's say positive, for example, to a room. And you emit it at a very, very high level. Otherwise, it wouldn't work. Like 100,000 per cubic centimeter or more, maybe 1 million per cubic centimeter. And then if you have a sufficient concentration of particles, they will acquire this ion. They will become unipolarly charged. They will repel, and they will end up in surfaces, or most of them will end up in surfaces. Now, we have evaluated this method in a 25 cubic meter chamber as well as we calculated the aerosol transport in this chamber, and then we found that this method may have a potential for removing particles not as high as filters, but the problem is that most of those emitters, ion emitters, they also emit ozone, and there was a lot of stuff said already about ozone. So ionic air purifier, the conclusion, low power consumption, they are silent, they have low operational cost, they have a possibility, unlike ozone generator, to remove airborne viruses, bacteria, and fungi in confined areas. The volume is a king here. If you go for a large volume, it wouldn't work. There are also wearable devices available utilizing the same method. Now, as I said, the CADR, clean air delivery rate, is not as high compared to filter-based air purifiers. The particle removed from air end up on indoor surfaces, so the hazard is not removed, essentially. It remains, it's just not in the air. And efficiency is time dependent. In other words, a certain startup time is needed to saturate the air volume with ions to expect a quantitative effect. But the main problem is, again, it's not for all ion emitters, but for most of them, those we happen to evaluate, that ozone emission is a problem. Now, speaking of ozone generator or ozone emission in indoor air, I can show you results of our study, just a few graphs. We actually using one of those devices in a 25 cubic meter exposure chamber. And you can see here the particle size distribution, the initial particle size distribution. It was ambient aerosol. The initial level of ozone was very low, 0.004 ppm. That's considered as a baseline. And then ozone, we pumped ozone in this chamber. So in a very, very slow way. So here you can see the increase to the level that in some environments may also be considered as a background level. And you see that this distribution didn't change much. Now, at the higher level of ozone, in 40 minutes, you see something started going on over here. These are small particles in nanoscale. And here in 60 minutes, when the ozone level is 0.073 ppm, you see this effect solidifies. And then later on, when the level goes up to 0.1 ppm, you see a pretty clear effect of the generation of ultrafine particles. And again, the mechanism has been discussed here today, so I will not talk about that. I have to say that the effect or similar effects have been discussed in earlier literature, specifically for ozone reaction with terpenes and other materials. Now, approach 2, we want to inactivate microorganisms. We don't care about concentration anymore. We just want to kill viable microorganisms. So there are a number of principles or number of methods which are used, including thermal exposure, ultraviolet irradiation, cold plasma. There are others, including ion emission, that some manufacturers claim that they are also good for microbial inactivation, ozone generation, and very recently the photocatalytic oxidation. Now, speaking of inactivation by heat, we ran this system where we had a specific temperature in the exposure chamber, and we ran the BIOAEROSOL flow through this chamber. This experiment specifically deals with BG spores, the simulant for Bacillus anthracis, the causative agent for anthrax. So this is a very hardy spores. And you can see here that inactivation factor, which is a measure of how well we can inactivate, goes up. Two curves represent two different flow rates, 18 liter per minute and 36 liter per minute through the system. You can see that this is a pretty steep exponential curve that may end up at about 350 degrees centigrade when 1 out of 100,000 spores will remain viable. So we concluded that thermal inactivation has a great potential. for indoor air cleaning as long as we can process enough air with this temperature, exposed to this temperature. So it will work even for stress-resistant bacteria. And we also concluded that because of this exponential trend, the concept of temperature dose is quite feasible. Now, a similar study was done about at the same time by our colleagues in Korea. They looked at the survival of fungal spores. We look at bacterial spores, they look at fungal spores, and they actually started digging in terms of a mechanism, what is behind of this thermal inactivation, specifically for fungal spores. So they worked with Aspergillus versicolor and Cladosporium cladosporioides. They actually compared the controls, and you can see here controls versus exposed. They concluded that the short-term exposure to high temperature changed the surface morphology of the aerosolized fungal spores, and they linked it with the mechanism, proving that it's quite feasible to expect that their inactivation will be very, very high, including close to 100% inactivation. Now, we started looking in a similar way what is going on when you run the particles through this exposure chamber in different temperatures. And actually, it was quite interesting because there are a number of mechanisms that may be behind this inactivation, because the spore itself is quite a complex creature. And when you look at the interaction between heat and the spore, you may speculate that there could be a DNA mutation, the protein damage, cell wall integrity issue. All these effects may be behind the thermal inactivation, but the bottom line that it's quite efficient. Now, a similar method was applied to airborne viruses, also thermal inactivation. The only thing that the viruses are more susceptible to heat. So you don't need to run it to 300 degrees centigrade really to see 100% inactivation. There was an interesting work, quite elegant work, done by Szymanski's group in University of Vienna, when they looked at the spectrum after transformation of an infectious virus to a non-infectious form, read viability. So what they found that the viral proteins were released from a virus at a certain temperature. And that was basically a method, it was a principle that they apply to a first prototype of an air cleaner that actually heated the viruses only to a temperature of 60 degrees centigrade. And it was enough given the time of a transport through the chamber was about 1 to 3 seconds. A few words about UV. Actually, there's a lot of studies now about the UV radiation and its effect on the viability in indoor air. The earlier studies were not motivated by aerosol mitigation. It was mostly for other purposes like food products, water purification. When we look at this, we have to acknowledge that there are differences in microbial inactivation that occurred in aerosol phase versus surfaces or in liquid, and they deal with an interaction between microorganisms and biocidals, access to oxygen and nutrients, and adaptation to environmental conditions. So, for example, one environmental condition, such as relative humidity, happened to impact the viability a great deal. So the inactivation rate by UV here was referred to UV irradiance and plotted against the relative humidity. It's work done by Mark Hernandez in his group, and you could see that the dependence on the relative humidity is very evident. There was another interesting work where UV was actually tested together with the heat effect, with the thermal effect. The work was done at Konkuk University in South Korea. And what is plotted in this graph, you see here just the heat effect. This is an activation rate, this is temperature. So green shows just the heat effect. UV is somewhere here, it's just at the room temperatures. And then red and black curves show the results when the thermal energy works in combination with the UV. The only difference is that here you have only the red corresponds to 1 UV unit and black to 2 UV units. So you can see that a certain synergy can be demonstrated here, which is very important. Now, I leave aside the fact that some of these UV lamps may generate ozone, and it was enough said about that. Now, atmospheric pressure cold plasma, something that started being investigated very recently. That's this work by Chinese colleagues from Peking University. You could see here the MS2 virus being exposed to or treated with the cold plasma. This is controlled, this is exposed. So images of these virions showed a difference in fragmentation. So the damage was observed to the viral RNA genes that are coded to express 4 different viral surface proteins. So the inactivation here occurred to be achievable up to 2 orders of magnitude for viruses, and we have no data for bacteria at this point. Now, inactivation of viable microorganisms may be done not only in the air, but actually when they're already collected by a filter. And there are different techniques which have been discovered and investigated, including filters with embedded iodine, irradiation-assisted filtration. Aluminum nanofibers were used as they were shown to strongly retain viral particles which are naturally negatively charged. There was an attempt, again, to combine the UV irradiation and a microwave radiation. So all of these techniques are still under investigation. Let me summarize the inactivation method part. The filtration may efficiently remove particles of biological origins, but it does not have an inactivation effect, except the situation when antimicrobial coating is used on filters. Electrostatic precipitation has not yet been extensively deployed for bioaerosol exposure mitigation, and there is no evidence of its very high biocidal potential. Again, it may collect particles, but not necessarily kill them. The ion emission does not produce a very high CADR, but requires no high energy or power consumption. It is silent and inexpensive. The big problem potentially here is generation of ozone. Heating may provide a high microbial inactivation for different classes of microorganisms. UV is efficient to inactivate airborne viruses and vegetative cells, but may not be that efficient when it comes to spores, and it depends on environmental condition. Now, new methods, cold plasma, recent tests showed that this method can effectively inactivate viruses, the same expected for bacteria, but no data is available. Ozone emission, I would say useless for both, for a decrease of the particle concentration in the air and for inactivating microorganisms. Photocatalytic oxidation, viability loss was identified, but the effect is not great, especially in large areas, and the same concern remains the byproducts. And microwave irradiation of filters is being now tested with respect to the inactivation. The last slide, as I mentioned, the same approach can be applied to respirators. It's also a way to reduce the exposure to biological aerosols. And essentially, we have done a lot of studies on respirators as well as our colleagues around the world. Essentially, what we can conclude that the conclusions made or findings for stationary filters may apply to respirator filters pretty well, except 2 specific areas. Protection may not be efficient because of the facial leakage. So here, the filter of the respirator may be good, but the facial leakage actually diminishes the effect. And the other thing is that the use of so-called antimicrobial respirator filters may be questioned for 2 reasons. There are 2 things are not known. The dermatological effect when it interacts with the skin, and inhaling of biocidals. In fact, they are released from this so-called antimicrobial filter. So I would stop at this point. Thank you.